where does vision start? The smiley face. The question is, why do we perceive a circle, two full stops and a closed parenthesis turned on its side as a face? And the answer is that as social mammals, you have cells in your temporal lobe that spend their whole time looking for faces in the visual field. And when they see a face, they light up and they go, face. And they are there because a face is the most important thing in your visual field today. Okay? If you see a face, it's going to be giving you important clues about the organism to which that face is attached. Yep. Wants to buy me a beer. Wants to beat me up for running away when I didn't buy him a beer. Wants to become my life partner. Is my life partner's parent. I should avoid this one. All of those things. Yep. Faces are the most important thing in your visual environment, and you have cells that go face. Oh, and then your attention blocks down on that face. Face. Looking at the face, okay? So whenever you're walking down the street and you see a face, your attention, your visual attention, is brought to that face to see what it's doing, what emotions is it showing, how's it interacting with me, how's that going to affect the way that I should behave. What I want to look at when we consider vision is to consider how those cells operate. How do the visual images that we see build together to give us those cells that recognize faces anywhere in the visual field? And by corollary, how do the other shapes that we recognize, beer glass, sandwich, all the important things, how do they build up in our visual field, and how do we get information about the visual environment? This is the neural network at the back of the eye. The eyeball is down that way. The choroid plexus and the sclera is up at the top. Light is coming up from the bottom, through the cells, hitting the choroid plexus and either being absorbed or reflected depending on how much incident light there is. And these are the cells that we're going to talk about to start with, to talk about how those cells construct the beginnings of the visual image and what information those cells carry up to the central nervous system, or rather these cells carry up to the central nervous system. You'll remember when I was talking about sympathetic ganglia that I said that synapses mean information processing. People remember that? Yeah? What we've got here are loads and loads of synapses. And so it follows that within the retina, we've got loads and loads of information processing. And so what comes out in these axons in the ganglion layer is not necessarily what you might think from the light that goes in and stimulates the rods and the cones. The other thing to say, and this is an important principle, is that the sensory transducers do not undergo action potentials. When you look at a sensory transducer, in general terms, and it is stimulated, it doesn't generate an action potential. It changes its membrane potential. And in fact, in the eye, the first cells in the visual pathway that generate action potentials are the cells in the ganglion layer that go on to form the axons of the optic nerve. So, transducers, rods and cones, do not generate action potentials. Bipolar cells do not generate excuse me, action potentials. Horizontal cells do not generate action potentials. Ganglion cells generate action potentials. So what's happening is we're getting a complex network of membrane potential changes in this nervous plexus, which is influencing the rate of action potential generation in those cells. We're going to come back to parts of this diagram and consider in more detail how those membrane potential changes affect action potential changes. But before we do that, I think it's a sensible idea to look at the concept of a visual field. What is a visual field? A visual field is the area of your vision which can affect the particular thing we're looking at. So we can say that a particular area of the optic cortex has a particular visual field, responds to light from a particular place in the visual area. And we can also say that every cell in the pathway between the transducers and the optic cortex and even onwards has a visual field. It has an area within the whole of the vision that it responds to. So I want to look now at the visual field of a ganglion cell. These are visual fields in the optic nerve, and what I want you to imagine is that the slide is an area of retina, and we are looking at one axon in the optic nerve, and we are drawing on the area of retina the area to which that nerve is capable of responding. And if we look at that, we find that there are two distinct sorts of axons. There are on-center and off-center. So an axon with an on-center visual field is facilitated by light landing in the center of its visual field or area of sensitivity, and is inhibited by light falling on the periphery. And conversely, an off-center visual field is inhibited by light landing on the center of its visual field and facilitated by light landing on its periphery. Seems a bit complex the first time you look at it, but it makes good sense as to why that happens. We'll talk about that later in the lecture. For the moment, I want to go back to this slide, and I want to say that it's even more complicated than that. Within the transducer layer, we've got rods and cones. Rods responding to low levels of light in a monochrome fashion, grayscale. Cones responding to coloured light. And there are three sorts of cones. The red cones respond to red light. The blue cones respond to blue light. The green cones respond to green light. Different wavelengths of light. So when we get to visual fields here, we've got 8 separate populations of visual images coming in. We've got on-center and off-center for rods and 3 populations of cones, all separate. And when we consider 8 populations, life just gets too difficult. So what we're going to do is we're going to talk about on-center and off-center briefly, and once we've got past that, we're just going to consider an on-center population down through the central nervous system. But you need to remember that every time we make a logical step, there are 7 other populations of parallel processes going on in the visual system. So a visual field is an area of the visual field to which a cell responds. And for the ganglion cells, it has this donut shape, and if it's on-center, there is facilitation in the middle, and there is inhibition at the edge. So let's talk about how those visual fields are generated. Here's a cone. Could be any sort of cone, could be a rod. It's a light transducer. Here is a graph of the cone's membrane potential at rest and when light shines on the cone and stimulates it. And you can see that when light stimulates the cone, the membrane potential reduces. I've put two bipolar cells taking information from that cone, and I'm going to pretend that one is an on-center bipolar cell and the other one an off-center bipolar cell, to illustrate the fact that cones are neither on-center nor off-center. They're just transducers. Light stimulates them, their membrane potential changes. Let's look at the on-center bipolar cell. You'll remember, I hope, from your basic neurophysiology that when you stimulate a cell, there are two options. There are excitatory postsynaptic potentials and inhibitory postsynaptic potentials. So stimulating a cell can either increase or decrease its membrane potential, and it depends which channels are opened on that receptive cell, either the sodium channels or the chloride channels. But for an on-center bipolar cell, hyperpolarization of the cone by stimulation with light causes a relative depolarization of the bipolar cell. Causes a relative depolarization of the ganglion cell, increases the rate of action potential generation by that ganglion cell. So that for an on-center visual field, stimulated in the center of its visual field, we get an increase in the rate of generation of action potentials, from a basal rate to an increased rate. Let's look at the other bipolar cell, this time an off-center bipolar cell. This time, hyperpolarization of the cone causes hyperpolarization of the bipolar cell, causes hyperpolarization of the ganglion cell, causes a decrease in the rate of action potential generation in the optic nerve. So we've demonstrated how stimulation at the center of a visual field causes facilitation or an increase in action potential rate in an on-center visual field, and inhibition or a decrease in action potential rate for an off-center visual field. What about the periphery of the visual field? How does that work? How does light shining in the periphery of a visual field have the opposite effect to light shining at the center? Here's a horizontal cell. The horizontal cell transmits information from peripheral cones or rods to those bipolar cells. But the horizontal cell has the opposite effect to the bipolar cell, so that when the cone hyperpolarizes, the horizontal cell of an on-center visual field hyperpolarizes, causes the ganglion cell to hyperpolarize, reduces the rate of action potential generation. And for an off-center visual field, the horizontal cell brings information in from the periphery. The cone depolarizes, that's the peripheral cone. The horizontal cell hyperpolarizes, but this time the off-center bipolar cell depolarizes, and we get an increase in the rate of generation of the optic nerve. So the take-home message here is that the horizontal cells are the converse of what's happening at the center. When we get facilitation at the center, an on-center visual field, we get inhibition in the periphery. When we get inhibition in the center, an off-center visual field, we get facilitation in the periphery. What we're looking at is the concept which allows this bipolar cell, say, to respond to light from this cone and also through a horizontal cell, some of these cones. And again, we've drawn a section through the retina here, but imagine this all going on in 3 dimensions. So imagine for this bipolar cell, there's a cone here, and one at the front, and one at the back, in a circle all around it, which are all forming the periphery of the visual field. When you take that retina and look at its surface, you take that slice and you turn it through 90 degrees, you get this visual field, such that an on-center visual field is facilitated by light falling on its center, but inhibited by light falling on its periphery. These visual fields with this donut shape, this facilitation and inhibition, or inhibition and facilitation, are central to our understanding of visual physiology and central to our understanding of sensory physiology generally, because they allow operation through a process known as lateral inhibition. Lateral inhibition is a feature of most, if not all, of the sensory systems. I want to consider another business entity. We'll move on from McDonald's this time, and we'll consider a weather forecasting service that operates over New Zealand. Now, if you were to set up in Wellington, say, a weather centre that knew what weather was going on all over New Zealand, you might set it up with a switchboard, and you might have people all over New Zealand whose job it was to phone up and say what the weather was, so that you knew what was going on centrally in Wellington. And then you can distribute that information wherever, well, wherever people pay for it, I suppose, but wherever you see fit. So let's imagine you want a 5-minute resolution of weather. And let's imagine you want a 20-kilometre resolution in a spatial sense. So every 20 kilometres throughout New Zealand, you have somebody with a telephone, and every 5 minutes, they phone you up to tell you what the weather is. How many people do you need on that switchboard in Wellington? 3 going to do it? No. And how many phone lines are you going to need? Lots, yeah? Okay. But many of those people are going to be telling you what you already know. It's Palmerston North again, it's still raining. Yeah? It's Wellington, the wind's still blowing. It's Auckland and we don't really know what's going on. Yeah? So a lot of the information that you get to your centre is wasted. It's repeated information. Let's have a different system. Same number of people around the country, same number of sensory receptors, but this time you say, hey, don't phone up every 5 minutes, we know you're doing your job. Just phone up when the weather changes. Yeah, but you're never going to hear from Wellington, are you? Still windy. But when the weather changes, people are going to phone you up and they're going to say, hey, the weather's changed here, it stopped raining, it started raining, whatever. Does that reduce the number of people you need on the telephone in Wellington to answer those calls? Yeah, hugely, because you're not getting calls from all of the sensory receptors every 5 minutes. You're getting calls from sensory receptors when something changes. Does that still give you the 5-minute temporal resolution that you need? Yeah, in fact, it's even better than that, because people don't just phone you up every 5 minutes. If you imagine with the 5-minute phone calls, you could phone up, say it's sunny, put the phone down, and 10 seconds later it starts raining, and Wellington doesn't get to hear about this rain for another 4 minutes and 50 seconds, because that's when the next phone call goes through. But if you sit there and wait till it starts raining, you can tell Wellington about it as soon as it started raining. So for fewer sensory processing units, you get the same spatial resolution and better temporal resolution. You've got a better system running cheaper. And that's what lateral inhibition is all about in the central nervous system. It gives you a more sensitive sensory system that is cheaper to maintain in terms of processes centrally than a system that's giving you the information all the time. So what happens is the central nervous system doesn't pay any attention to neurons that are sending in action potentials at the same old rate. Doesn't pay any heed at all. It's only when that rate changes that the central nervous system pays attention to that information. And so the central nervous system focuses its attention on things that change. Let's go into a room that you've never been into before, with your eyes closed. Open your eyes when you get in the room. Get a friend to help you into the room, because it can be kind of embarrassing if it's a public space, and But walk into the room with your eyes closed. Open your eyes, count to 5, close your eyes again, and think to yourself what you remember of that room. And what you'll remember is, you'll see the doors and the exits and the bright lights. You'll see where the walls are. You'll see where the bar is in the corner or whatever. You'll see where the gents are. You'll see the big things, but you won't see the detail. You probably won't notice the pattern in the carpet, or what kind of curtains there are at the window, or exactly how many people are sitting at that table, because those details you'll fill in later. A slightly different test. Go into one of your friends' rooms if you're flatting, or one of your family members' rooms if you live at home, and take something from the room, from off a shelf or something, and just quietly put it somewhere else. And see how long it is until they notice. Because when they go into that room that they've been in before, that they're familiar with, they don't look all around the room to see what's going on. They load into their mind their memory of what that room looks like. And they gradually update it and keep it current. But they don't put all the little ornaments in the right place on the shelf by looking every time they go in. They remember that that tacky ornament was there on the shelf. And it might be an hour or 2 hours or 3 weeks before they notice that it's actually not there anymore. Okay? It's another way of reducing visual processing, looking for the differences in the environment and focusing on them. So let's look at how lateral inhibition develops and how it saves us processing. Here is an area of skin this time. Looking down on the area of skin, these are Pacinian corpuscles within the skin, touch receptors. And I've drawn them in regular rows because then it's easier to draw a little hexagon around them and say that is the visual field of that, or the receptive field rather, of that Pacinian corpuscle. This hexagon represents the area of skin over which that Pacinian corpuscle is sensitive. And then we can just draw the hexagons and everybody's happy that my histological skills aren't being tested. So let's imagine an area of skin with conjoining sensory receptors, like so. Here it is, and each of those corpuscles in those receptive fields is generating its baseline rate of action potentials, and the central nervous system is taking no notice. And here it is, it's this kind of sickly pastel pink colour midway between white and red. A baseline standard rate of action potential generation. Let's stimulate one of those receptive fields and give it a little tickle in the middle there. When we tickle this receptive field and only this receptive field, what's going to happen to the rate of action potential generation in those receptive fields? The answer is that the rate of action potential generation centrally is increased, and the rate of action potential generation in the receptive fields immediately adjacent to it are decreased, facilitation and inhibition. Where have we seen a receptive field that looks like that before this morning? Looks like the receptive field of the ganglion cells? Yeah, same sort of donut affair. It's exactly the same physiological principle. With touch, it happens in different cells, in different parts of the sensory pathways, but the principle is exactly the same. And so when we stimulate one receptive field, the action potential rate generation changes in 7 receptive fields. How does that save us anything? Well, let's look at what happens when we stimulate receptive fields over a larger area. And let's build up a picture of how the rate of action potential generation is altered when we tickle over a larger area. So this is what's happening. Let's consider this receptor here. It's being facilitated because it's being stimulated, but it's also being inhibited by all those around it. This one here is being stimulated and so facilitated but inhibited by every receptive field around it. This one here is not being stimulated and is being inhibited by stimulation from 3 adjacent receptive fields. And so if we take the T's out of the way so that you can see the colours more easily, you can see that we've got this ring of facilitation surrounded by a ring of inhibition. And that in the centre of the area of stimulation, the rate of action potential generation is the same as in an area that's not being stimulated, an area distant from the area of stimulation. And so what this is doing is it's focusing the attention of the visual, the sensory modality on the edges of that area of stimulation. And that is lateral inhibition. It's a good thing because for a large area of stimulation, the periphery of that area involves fewer sensory receptors than the area of the stimulation. And so it gives us accurate information about changes with as few as possible sensory cells sending that information to the central nervous system, giving us a reduced number of processing units that need to deal with that information, giving us better efficiency. It also gives us an increase in spatial separation. I'm not going to go through this morning, but if you have a look in Kandel, Schwartz and Jessel, you will see that there is a box there under the skin senses dealing with lateral inhibition, talking about how if you take 2 stimulating areas, and it uses 2 pins, and move them progressively closer together, there comes a time when they're so close together that they are sensed as just one area of stimulation. And it's called the spatial resolution of sense. A system that deals with lateral inhibition can distinguish 2 separate sensory stimuli when they're closer together than a system that doesn't deal with lateral inhibition. And so it's giving us, for less processing power, a system that is more spatially sensitive. If you want to see some examples of lateral inhibition, take a hot bath and jump into it. Where does it feel hottest? Yep, a ring round the top, right where your ankles are in the water. That's where it feels hottest, right at the surface. Not because it is any hotter there, but because that's the area of boundary between the area that's being stimulated, the heat receptors that are being stimulated, and the heat receptors that are not being stimulated. Same if you're a poor student, you can't afford a hot bath if you place your hand in a bucket of cold water. It feels coldest, not at your fingertips, which are deepest under the water, but in a ring around your wrist or forearm, which is where the boundary is between the cold water and the environment. And the advantages of lateral inhibition: it reduces sensory neural torque, and it focuses on areas of spatial contrast. This is most of the brain of a dog. It's a brain stem from which I've dissected off the cerebellum and the left cerebral hemisphere and left the right cerebral hemisphere and the brain stem intact. Down at the bottom here you can see an optic chiasma, and you can imagine that there might have been 2 eyeballs there once. What I want to do is I want to draw the connections between the retina and the optic cortex onto this diagram and demonstrate the ways that the sensory nerves travel into the central nervous system. here is the ganglion cell axon travelling from the eyeball to the optic chiasma and then in to the ventral surface of the brain stem. A single synapse on the way to the visual cortex in the lateral genicular nucleus. And that is a basic representation of the visual pathway. There are a few complexities that we need to put onto that pathway in order to fully understand it. And the first complexity that I want to mention is the optic chiasma itself. Some of the neurons in the optic chiasma decussate, means they cross the midline, so they go straight across from, say, the right eye to the left side of the brainstem. Some of the neurons do not decussate, do not cross the midline. And that brings us to decussation as another one of those important neurophysiological concepts. In general terms, neurological pathways decussate. At some point, they cross the midline. And some of them multiply decussate. They cross the midline and then they cross the midline back again. But almost all of the neurological pathways cross from one side of the body to the other side of the brain. And in fact, it's so unusual not to cross that I can think of examples of neurological pathways that don't cross on the fingers of one finger. And we'll meet that pathway next semester. Let's talk about decussation in terms of vision then. And let's look first of all at the visual pathways of a typical herbivore or prey animal. Herbivores tend to have one eye on each side of their head, looking out sideways, and they tend to have a wide field of view. They can see most of the way around. They pay for that because they don't have a well-developed stereoscopic area. They don't have an area that both of their eyes are looking into that gives them a good idea of depth perception. So any of you that have tried to creep up on a sheep from behind will know two things. One, that the sheep can see you even if you approach from behind. And two, because it doesn't have a good idea of how far away you are from seeing you, it will spook sometimes even when you're quite a long way off. If we look at the visual pathways in the optic chiasma of the herbivore, we get this picture with almost all of the nerve fibers decussating. And the concept that I want you to get here is that information in the left visual field is dealt with by the right optic cortex, and information in the right visual field is dealt with by the left optic cortex. If we look at the kind of animal that's likely to be sneaking up on the sheep, a carnivore or prey animal, then both the eyes face in the same direction, forwards usually. And that means that typical carnivores don't have a very wide field of view. They have to turn their heads round to be able to see widely around them. But within their field of view, they can bring both of their eyes to bear on an object from a slightly different perspective, and the difference in the two images can be processed to give them an idea of depth. Here's the visual pathway of a carnivore. And here you can see that some of the neurons in the optic chiasma decussate, some of the neurons in the optic chiasma don't decussate. So what's happening? Well, how efficient would it be if both eyes, looking at the same thing, sent information to their own optic cortex, and each cortex had to deal separately with information on the same thing? Would that be efficient neurological processing? No, because we've got a duplicated system there, and also these two structures are a distance away from each other. They're going to have to communicate with each other. It's going to slow things down. So the system that's built here preserves the left optic field on the right and the right optic field on the left. And what happens is neurons that are dealing with information in the left optic field, whether they be in the left eye or the right eye, end up in the right cortex. So if they get into the left eye, they decussate, and if they get into the right eye, they don't decussate. But the information has decussated. It's either decussated in the optic chiasma, or it's decussated coming across the animal's midline before it gets into the eyes and into the neurological system. So that each cortex is now dealing with the same half of the visual field. And it's quite interesting. If you deal with a carnivore that has a lesion in its optic nerve before the optic chiasma, they lose sight from one eye. But they can still see just as well with the other eye. But if you deal with a carnivore that has a lesion in its optic pathway after the optic chiasma, it can still see from both eyes, but it can only see in one hemisphere, one side of its body. So it will lose sight from, say, the left side of its body in either eye, but keep sight from the right side of its body in both eyes. And it's because the pathways at that point have sorted themselves out into right visual field and left visual field, and all the information has crossed the midline. So just to revise that, if a herbivore sees a strange little man in its right visual field, all the information goes into its right eye. The information decussates in the optic chiasma, and the image of the strange little man is dealt with in the left or the opposite optic cortex. If a carnivore sees an image of a strange little man, the information goes to both eyes. And you can see that on the ipsilateral eye, the information is hitting the medial retina, and in the contralateral eye, it's hitting the lateral retina, because it's aside from the midline. Because of the decussation, i.e. the medial retinal ganglion cells tend to decussate and the lateral retinal ganglion cells tend not to decussate, the image of the strange little man is constructed in the one hemisphere that deals with not information from one eye, but information from one visual field, so that the whole thing is efficiently put together. So what I want to do now, having spoken about where the neurons are, introduced the concept of visual fields last time, and spoken about the visual fields of the optic cortex as a whole, I now want to go and look at the visual fields of the individual neurons and look at how they change. So this is a diagram of the visual fields of those first two neurons that I showed you, the ganglion cell and the concentric cell. And you'll see that the ganglion cell and the concentric cell have the same visual field. Okay, what do synapses mean? Processing. Yep, processing and slowing down. Good. How much processing is going on to generate that lower visual field from the upper visual field? None at all. They're the same. Yeah? So why have a synapse in a pathway if there's no processing? The answer is that in the visual pathway, the conscious visual pathway, there is no processing between the ganglion cell and the concentric cell. But there is processing that goes on at this level, and you'll remember that that synapse is in the brainstem. There is processing at that level, but it's processing that illustrates another neurological concept. And the concept we're going to talk about this time is divergence. In fact, two concepts, divergence and convergence. Divergence is the concept of information from one neurological pathway being diverged to lots of destinations. And convergence is information from lots of different neurological pathways converging on one neurological pathway. And we'll find out next semester that divergence is a feature of sensory systems and convergence is a feature of motor systems. And so when we look in more detail at what's going on in the geniculate nuclei and also the colliculi, we see that the processing that's going on at that level is all about divergence. This diagram is in your notes, and all of it's in your notes. It's also in Tony King's book, and it illustrates the processing that goes on at that level. The axon in red coming through the optic chiasma is the ganglion cell. The axon in blue, leaving the lateral geniculate nucleus and going to the visual cortex, is the concentric cell. And you can see nothing much happens there. But if you look at what's going on in the rostral colliculus, you'll see that the information from the ganglion cell is diverging into lots of different neurological pathways. And the information is going to where it's needed. So, the information that's going to the visual cortex is giving you conscious visual perception. It's going later on to build a visual image, to build an image of the environment. The information that's dealt with by the rostral colliculus and diverged or dished out to other pathways deals with the other functions of vision. Some of them you'll know about already, some of them you probably won't. For example, there is a pathway here to the parasympathetic nucleus of the third cranial nerve, the Edinger-Westphal nucleus, as it was called in the prehistoric days when I did anatomy. That nucleus controls the degree of dilatation of the pupil. And it takes information from the optic nerve about how much light is getting into the eye and uses that to set the degree of dilatation of the pupil to regulate the amount of light that's getting into the eye. So it's a closed feedback, a closed negative feedback loop. More light gets into the eye, the pupil constricts so that less eye gets in, less light gets in, and everything sorts itself out. What else does the eye do that we're not really conscious of? Well, you'll know that as you move through space, the eye fixes on various points in space and moves in jumps called saccades. And that also it's very good at fixing on the horizon to give you a concept of position in space. So there is a direct output from the rostral colliculus, and it's here, to the motor nuclei of the third, fourth, and sixth cranial nerves. So without going through the conscious centers, the information coming up from the optic nerve is being used by the motor system to fix the eyes in space. Another reflex, the visual image moves and the muscles of the eye move to keep the eye stabilized in space so that it can stay fixed on one object even though the animal that owns it is moving around. The tectospinal tract is a spinal pathway that's involved with posture. And so there's a direct feed from the visual image to the postural muscles. So that if, for example, the visual image slews this way, The postural system interprets that the animal is slewing that way, and as long as more than about 5 pints of alcohol are not involved, then the animal makes a postural adjustment and keeps itself still in space. It's a postural output, and it doesn't require conscious visual processing. The cerebellar cortex, again involved in regulation of the motor system, again taking a direct feed from those ganglion cells. And finally, the ascending and descending reticular formation involved in arousal and awareness and wakefulness. How easy is it to doze off in a disco with all the lights flashing? Not easy, because visual information is coming straight into the ascending reticular formation saying, hey, flashing lights, lots of noise, lots of things, you should be awake. How easy is it to stay awake in a lecture with the lights dimmed? Not easy, because the reticular formation is saying, oh, it's nice and dark, it's nice and peaceful, it's still only 8:20, you should be in bed. Yeah, okay. The visual pathways and the other sensory pathways are feeding into this arousal system and regulating the state of awareness. Again, if we look at lesions in these pathways, it's quite possible for an animal to have a lesion here or later in the visual pathway and to be unable to see, but still able to regulate its pupillary diameter in response to changing light, to move its eyes in response to the head or the body moving, and to make postural adjustments based on visual stimuli, even though the animal's blind, because it cannot construct a visual image because its conscious centers have been deprived of that information. So, the visual field between the ganglion cell and the concentric cell doesn't change, but there's a whole lot more stuff going on behind the scenes which is involved in diverging the information to lots of other sensory pathways, sending the information to lots of other places it's useful. The concentric cell delivers the information to the optic cortex, and then things start to get interesting, because then the optic cortex starts to build those visual fields into more complex visual fields. Here are the next two cells in the visual pathway, and these cells are both in the optic cortex. The simple cell and the complex cell, both cells of the optic cortex. We've got multiple concentric cells feeding into individual simple cells. And one concentric cell does not feed into just one simple cell. One concentric cell may feed into many simple cells. And at the same time, one simple cell will be taking information from many concentric cells. And so there's an information exchange such that, and here's an example, the visual field of the simple cell is a line with a particular orientation at a particular point in the visual field. So when you see a line, like the side of the door there, it's in a particular part of your visual field, it's got a particular orientation, there is one simple cell going, hey, I see a line, I've got one, me, I can see it. And it's taking all the information from the concentric cells and adding them together. It's an AND function. If it gets stimulated by all of its concentric cells, it says, oh look, all those little donuts added together in that thing, that's a line, that's me. The complex cell, the next cell in the pathway, takes information from a number of simple cells. But this time it's an OR function. And so when you add together all those vertical lines in a slightly different place with an OR, so that if any simple cell attached to that concentric cell is stimulated, then that complex cell is stimulated, you end up with a vertical line in a particular area of the visual field. So the concentric cell says, I see an area of light contrast here. The simple cell says, I see the summation of various light contrasts. It's a line in this orientation. And the complex cell says, hey, somewhere in this block of visual field, there's a line going that way. Now, if you could imagine that we would follow that down through about 7 generations more complexity, we would have a cell that responded to a particular shape for important shapes in the visual field. And the one that I'm thinking of is the one that we started with, the face cell, or the face cells. The cells in the temporal lobe that add together the inputs of various complex cells and various simple cells in various different orientations and say that combination of shapes in that relationship to each other is a face. And I need to look at that face and see what's going on in more detail. So can you see how simple visual fields are being glued together to give more complex visual fields and give more detailed information about the visual environment? This is a diagram of the surface of the visual cortex of a monkey. And I forget what species of monkey. But it's been shaded by the orientation of the simple cells in that area of cortex. So you can see that the lines across the top are a code for the shading. And so if we have an area here in red, that area contains simple cells which are coding for horizontal lines. And if you look at an area in green, that area is coding for information for vertical simple cells. If you look at the diagram, you can see that in many places over the surface of the optic cortex, there are areas where the colours kind of swirl around each other and come to a point. And one of them is coloured in white here. Can you see how if you go round that point, you get all the colours represented? And that there are lots of other points there. Each of those points represents the centre of an area of optic cortex that's coding information for one part of the visual field. So that this one might be coding for an area near the exit sign, and the one next to it is coding for the bit of wall just next to the exit sign, and the one underneath it is coding for the door just under there. Okay? So those are dealing with different small blocks of the visual field. And the visual cortex is arranged into columns, usually called barrels of cells. And each barrel of cell contains many simple cells, many complex cells, with lots of interconnections between them, which are responsible for deciding what's going on in one small area of the visual field. Each barrel contains between 20,000 and 100,000 neurons with their interconnections, and their job is to work out what's going on just there, and to feed that information on to more complex cells in such a way that those complex cells can say, well, there's a big red thing there, and a sign that says wait there, and a rain shelter there, that's a London bus, I'll go and catch it. Okay? It's interpreting the visual information in a meaningful way for the animal's environment. The degree of complexity in the mammalian brain is greater than the coding precision of the mammalian genome. And that's a complicated way of saying that you couldn't write down in your genomes all the information that you need to build your brains. There just isn't enough room. How do we get this degree of complexity into the adult brain when we can't code for it in the genome? And extending that to other sensory systems, how do we get the degree of complexity that we have in other sensory systems into our brains when we can't code for it in the genome? Well, the answer is that the brain is not a solely genetic construct. It is an interaction between the genome and the environment of the organism throughout its life. And there's a very elegant, but as I was discussing earlier, slightly horrific paper that was published in Nature some time ago, which looks at that interaction between the genome and the environment. So here is a diagram illustrating the average number of connections per cell in the optic cortex during the time after birth in days in a kitten. And you can see that in the early days after birth, there is an increase in the number of connections in the optic cortex. The complexity of the structure is increasing. But in the yellow period, when the eyes open, and immediately following the yellow period, there is a huge increase in the complexity of the optic cortex, which follows the first sensation of light after the eyes open. This is the same diagram, except this time the kitten didn't get to open its eyes because they'd been sutured closed, preventing it from opening them. And so you can see that not only does that leap in complexity not happen, but the degree of complexity in the optic cortex actually decreases. And if you wait until this point and then allow the eyes to open, you don't get that increase in complexity. Something in the developmental process is saying, hey, now is the time for the optic system to be sensitive to the environment. Let's build some connections. And at that time, the eyes are opening, the information's getting in, the connections increase. The animal is interpreting its environment. But if you give that information to the animal, that processing information, you say, hey, time to interpret the environment, and the environment's black, there's no sensory input, the cortex goes, nothing to see, and it withers away to some extent. That's interesting in all sorts of ways. I want to mention two of them, and the first one I want to mention is the alterations that these studies caused in clinical medicine. There are various conditions where babies are born with relatively normal eyes but unable to see. Congenital cataracts is one of them. And the traditional management of those cases was to wait till they were, say, 3 years old and then do surgery to take the cataracts out so that they could see. Because, hey, don't need your eyes till you're 3, really. And a newborn baby is a much greater anaesthetic risk than a 3-year-old infant. So let's wait a while and let's do the surgery when the baby's bigger, stronger, healthier, easier to do the anaesthesia. And yeah, those children could see, but they could never see really well. They could never see as well as their counterparts who didn't have those diseases. So hey, that's explainable, isn't it? We gave them some sight. They started with none, we gave them some. That's a success, and maybe their visual pathways were a bit damaged anyway because there's all this damage going on. After this study was launched, or was publicised, medical management of these children changed to the point that now they have their cataracts removed in the first week of life, before they start to do this visual processing. And now as young adults, those children see as well as their counterparts. They have no visual damage residual from their blindness at birth. And it's because they could see at the time when their visual pathways are beginning to interpret the environment. So you need the visual information to make those pathways grow. The second example, well, it's two examples, but it's to the same thing, is what happens if you're in an abnormal environment when you get to that stage of development. It's been demonstrated that if you raise monkeys in an environment where they never see a straight line and where all the lines are wiggly, then the complex cells and the simple cells in their visual cortex don't code for straight lines, they code for wiggly lines. And they're looking for wiggly lines in the environment because that's what they could see when the cortex was developing. And if you raise monkeys that never see another monkey's face, they don't develop those cells in the temporal lobe that look for faces. Why would you? Nothing to see. Nothing in this environment that's going to feed me, give me milk, look after me, be empathetic to me. And those monkeys find it very difficult when they're reintroduced to monkey society to cope, because they don't see faces, they don't attach importance to faces, they don't see the emotion of the other monkeys around them, they're not empathetic to them. The alpha male comes along and beats them up, they don't have a nice time. So because they're not in a normal environment, when they're developing their cortex doesn't develop in a normal way, and that has consequences in later life. Another two examples of that, one a good thing and one a less good thing. People that are born blind and cannot have their sight restored for whatever reason will often be very good at spatial processing from sound stimuli. So they will hear something go on in a room and they will say, oh, it happened over there, in the same way that we will see something and say it happened over there. When they do that, they're using their visual cortex, in inverted commas. They're using the cortex that you guys use when you see something to process that auditory information. But it's become part of their auditory cortex. It's processing auditory information in a spatial way, in the same spatial way that an ordinary person that can see will process visual information. People that become blind after birth can't do that. They never do that because their visual cortices have already developed and already been locked into vision. And now they can't get those auditory stimuli into that area of cortex to do that spatial calculation and spatial reasoning. So if you're going to be blind from an early age, better to be born blind and to be able to use your visual pathways for other spatially related processing than to lose your sight at an early age. Last example, a completely different sensation, the sensation of pain this time. Over the last 30 or 40 years, medical science has got much better at getting babies that are premature and keeping them alive and turning them into adults. And we're pushing back the boundaries of that all the time, and we're now at a stage where if you're born at about 24 weeks of gestation, you've got a reasonable chance of making it. The problem is that the human pain pathways appear to undergo this processing at a late stage of gestation. And in the uterus, it's not a surprise to you, or it shouldn't be a surprise to you to learn that the normal environment is no pain, because it's not painful in a uterus. If you're in a special care baby unit, and 3 times a day you're having your trachea sucked out, and 5 times a day you're having a needle prick for a blood sample, and people are touching you, and there's sort of harsh material around you, and you've got tubes in your nose and all this sort of thing, that's not an environment that's associated with no pain. And so babies that go through that environment appear to have abnormally developed pain pathways. The pain pathways appear to be processing abnormal information. And it's now becoming more and more apparent that people that have a history of multiple traumatic or multiple noxious stimuli in the environment of a special care baby unit as premature babies have an increased risk of psychological disorders as young adults. Increased risk of depression, increased risk of schizophrenia and bipolar disorder, increased risk of suicide as young adults. So their abnormal pain processing at that stage of their development is building an abnormal central nervous system that is with them for the rest of their lives. So what I'm trying to say here is that your central nervous systems are not just something that you're born with and there they are. They're something that develop because of the environment that you're in. And they are processing machines. They develop and process the environment that you give them. And if they're given a normal environment, or what we would call a normal environment, and who knows what that would be, they develop in a normal way, and who knows what that is. But if they are deprived of something or given something abnormal at critical times in their development, they develop in an abnormal way and become a different processing machine because the environment that they're trying to interpret is not what we would call the normal environment, and so they get good at interpreting that abnormal environment.